Yeah, I'm going to present you today my current project about detecting a pangenome score. So yeah, nowadays we do have many individual genomes available per species for many species. And yeah, if we compare these individual genomes, then we encounter a lot of differences in the genomes. And in particular, if we compare bacterial individuals from bacterial species, we can even encounter differences in their gene contents. And so here, question that arises is, so if different individuals from the same species are so, yeah, diverse, what actually defines a species on the genomic level? So this question can be answered by a classical pangenomics approach. Let's assume a genome is a set of strings over the DNA alphabet. Then we say that we can say that a gene-based pangenome is the set of all the genes of the individual genomes. And a core genome is then all the shared genes shared by all the individual genomes. Yeah, considering the pangenomic score is important for many applications. On the one hand, it allows us to study the genetic diversity of a species in depth, but also it has some medical relevance. For example, when it comes to drug development or vaccine design. And also it has applications in crop plant breeding. But there are also some drawbacks of the gene-based pangenomic approach. We have, on the one hand, we have a very expensive pre-processing. We need to assemble all the genomes that we want to consider, and also we need gene annotations for them. Then we need to map the genes to each other, which means we have to identify the homologs. And also, if we only consider the genes, then we cannot find any core features that are below the gene level. And also we cannot find any features above. Therefore, one could also move to the sequence-based pangenome. And the sequence-based pangenome is simply the set of all the individual genomes, so all the sequences. And how to realize this? Well, for example, we could use a graphical representation. For example, a color-to-brown graph. But if we have such a graphical pangenome, sequence-based, how can we now define a core? That's the question. One goal that we can have here is that we would like to have a generalization of the gene-based approach, which means that we would like to have in our sequence-based pangenome also all the genes that we would find in a gene-based core. And one idea that we can have, what we can do here is to take all the k-mers as core, which are shared between all the input genomes. But then we will soon encounter a problem. And this I tried to depict by this multiple sequence alignment here. So what we see is, we see a multiple sequence alignment of some genes, which are homologs in different individuals of the same species. And these genes we might want to have as part of our core genome. But if we look in the sequence, then we see that actually the sequences of these genes do vary quite a lot. And this is the problem. Variation also frequently in core sequences. And if we only consider k-mers that are shared between all input genomes, we will not be able to find these genes as complete subsequences in our core.
Sorry to interrupt you.
Yeah.
But the presentation is becoming corrupt. I mean, we see black boxes here and there. So may you please stop screen sharing and restart it and to see if it becomes easier to read.
Yeah.
Please try share again now.
Okay. So does it work?
Let's. Well, we for example, the consensus is entirely black for me. I don't know what it is.
Oh, well, there's like there's some bars which are black, but below there should
yes.
be.
Okay, no, that's fine, but I can also see a gray box where you have the graphical pangenome question mark. Yes, so there are some artifacts from previous slides.
Oh, okay.
So maybe can you stop your presentation, the PDF viewer or whatever, and restart it and see if it's better?
Yeah, sure.
Yes, because now it's fine. Okay, but okay.
Let me just.
Yes, because.
Start it again. Okay.
No, because there are still some. Let's see. No, we have some. Well, we have some artifacts from previous slides. So, so you have the title of the box, definition core Kamer, and on the right we can see some graphical pangenome that comes from previous slides.
Okay.
Okay. Which is weird.
Let me try if I can just use a different PDF viewer.
Yes, try to use a different PDF viewer and see if it works.
Let's see. Okay.
And somebody suggests to share the application window instead of the entire screen.
Okay. Yeah.
That's a good suggestion.
Well, let's first try this.
Yeah, sure.
So,
try to.
Yeah, in case you see any artifacts.
No, still some artifacts.
Okay. So then.
So maybe try to share only the application.
Okay, how's it now?
No, I can't see anything. Okay, you started the presentation. Yes, no, now that's fine. That's fine, thanks, and go ahead.
Okay.
Thanks.
All right, thanks. Okay, so where was I? Yeah, this is what we had. So, okay, so our idea of how to define the sequence-based core is basically consisting of two different sets of k-mers. So the first one is defined here, given a pangenome that is represented as a colored brown graph, and we have two integers here, q and delta. Then we call a k-mer a core k-mer if it is present in at least q of the individual genomes. Okay, so in this example here, this would be true for these two k-mers over here and the one over here, TTA. Okay, and the second set of k-mers that belongs to our core is the set of bridging k-mers. The k-mer is called bridging if it lies on a path that connects two core k-mers, and the path should not be longer than delta plus the two core k-mers. So in our example here, this would be true for the green path here, connecting C, GA, and TTA, and the blue one here. Yeah, and that's it. So these two sets of k-mers should define our core. Here's the theoretical problem that we try to solve. This now has some advantages. For example, we do not need any assembly. We can just use raw read data here. Also, we don't need any annotations because we do not directly consider genes. And because we do not directly have to handle genes, we also do not need to do any gene mapping. But how can we actually find such a core? So first of all, for the set of core k-mers, this is quite easy. We just have to iterate over the graph, and for each k-mer count the number of colors it has. So that's quite easy. But for the bridging k-mers, this is a little bit more challenging. What we could do is we could say for each core k-mer, let's explore our graph by a breadth first search of depth delta, and each time if we find a core k-mer during this traversal, we just mark all the k-mers on the path as bridging. The problem here is that the search space grows exponentially with delta, and if the graph is complex, this can become quite probable, a kind of a problem very fast, let's say. So to improve this situation, what we could do instead is not to start the path traversal at the beginning of a path, at a core k-mer, but in the middle of a path. And then we would have to do two BFSs to both sides from the middle, but both have only a depth of delta over 2. So the search space is a lot smaller here. However, we need to do this for every non-core k-mer. So in the middle of such a path, there cannot be a core k-mer, but we have to consider all non-core k-mers here, and these can potentially be a lot. We can improve a bit if we use a compacted color-to-brown graph here. In a compacted color-to-brown graph, all the k-mers that lie on a simple path, on a simple non-branching path, are connected to a single node, and the node is called a unitic. And now if we use such a graph, then we only need to do the BFSs for the unitics. And usually there are a lot less unitics in the graph than there are k-mers. So we can save on the number of searches that we have to do here. Further speed we can gain if we store some information that we have gained during our searches. And we can use this information, for example, then to end future BFSs earlier, before we have reached the maximum depth of delta. We implemented this algorithm with Bifrost as the graph data structure, the data structure for the compacted de Bruijn graph, and the implementation is called Cora. Okay, so how does it perform? For this, we have done an evaluation with different pangenome data sets. One that I would like to present here is a Yersinia pestis data set of 48 strains in total, and an Enterococcus faecium data set of 153 genomes in total. Both data sets were tested for different parameters, for different values of k. As q, we always take n, so like every core k-mer has to appear in every single individual genome of the pangenome, and we tested a range of delta values. So, and of course, all the computations were performed on our cluster here in Bielefeld, single-threaded. So let's first have a look on runtime and memory usage. What we can see here is, first of all, that we need a little bit more time for the Yersinia pestis data set than for the Enterococcus data set. And we also see that runtime and also memory slightly increases for an increasing delta, and at some point this becomes very large, both runtime and memory, for one of the data sets. And this will also be something that we can discover for the other data sets for larger values of data. It's just not shown here. So, and this is because at some point the search space still explodes, even though we have this improved traversal strategy. We cannot do this for arbitrarily large data values. The question is, how large is data, or is it large enough? Like, can we detect a reasonable core here if we only use delta lower than 300? Maybe one note for the memory usage as well. So why is the memory usage actually so high? Well, if we do this BFS, we need to store a lot of paths if we have a high depth, and this is where the huge memory demand is coming from. Okay, so to answer the question whether we can already find a reasonable core, let's have a look on the results. So here I compared our implementation against another tool, Edgar. Edgar is a gene-based approach, so it detects the gene-based core genome. And the way I compared both tools is that I took all the gene annotations and I tried to detect the genes in our results. And of course, what Edgar outputs is already the genes. And then I calculated intersections between both results. So what can we see? First of all, we see if we don't use any delta, for this data set, our result is already not too bad. So we have a quite large overlap between the gene-based approach and our approach. But we can still improve a lot if we only use a small delta value here. So if we look on the number of genes that, for example, Edgar reports as to belong to the core, and that we do not find, this number goes down a lot as soon as we use a delta value. And to increase delta a lot more is not necessary here, so we won't improve our results anymore, or not really, at least. Also, we can see here that we find a lot of genes that are unique to our implementation. So there's a high number of genes that Edgar does not predict as to belong to the core, and this is mainly because Edgar does not consider paralogous genes. So as soon as there is a gene duplication in one of the individual genomes, only one of the two copies is considered for the core detection, and the other one is not considered by Edgar. This is, of course, something that we can do. We can also consider paralogous genes, and this is why we can find much more genes here. If we have a look on the other data set, then here's the results do not look so good. So again, we can improve if we start to use a delta value, but here we actually need a lot larger delta value in order to get a reasonable good overlap between both tools. So this the data that is sufficient here seems to depend highly on the data set that we actually do consider. Okay, and also we again have a lot of unique genes in this solution. So the next step now would be to compare our result to compare our implementation to actually approaches that do also predict a gene-based core genome. And this comparison is not performed yet, but I'm working on it. I would like to perform this comparison against Pansec, which is a tool that calculates the sequence-based core based on alignments. Then there is Sibylla by Ilya Minke and colleagues. And this approach also uses the Brown graph for the core detection. We have SplitMem. SplitMem even uses a compacted colored, not color, but a compacted Brown graph. And then there is an approach, IMGE, and they actually do not predict the core, but they do predict the accessory genome. So the accessory genome is not the amount of, all the parts of the pangenome that are shared between all the individuals, but only of subsets. And there are potentially more tools to compare against, so my literature review is not finished at this point. Okay, let's come to a conclusion, to some outlook, future directions of work. So first of all, I would like to generalize our model that we use for defining the core of a pangenome, also to allow for detecting an accessory genome. That should be possible only with slight modifications already. What might be also possible is if we use a range of delta values for our core detection, then we might be able to find something like a hierarchical core genome representation. This is kind of an idea that also Sibylla follows. Last but not least, if we have find the core genome, then we could also think about to generate something like a reference core genome of a pangenome. Okay, that's it from my side. Thank you for your attention, and I'm happy to take questions.
Thank you very much. So there are already 2 questions. Slide number 8. Why 2 breadth-first searches from the middle of the path? Could not you perform a BFS of size delta over 2 from the core k-mers and mark all the k-mer reached? The second time you reach a marked k-mer, it should be one of interest.
That's an approach that is potentially possible. So we could do this delta over k depth search from the k-mers, from the core k-mers, and then just mark the unitics where we can get to. However, we would also have to mark all the k-mers on the way, because it could be that like, I mean, no, we don't have to. Oh wait, no, we don't have to mark them on the way. It's okay if we just mark them at the border, but we also, if we are not able to reach from the other side, from the other core k-mer, if we are not able to reach these unitics again, we would have to unmark them all again, and then we would have to have another traversal. I mean, it could be that this is still an approach to go for. To be honest, I haven't tried that out, so it might work. Actually, nice idea.
Okay. The second question is, could we resume what you do as iterate over all bubble and super bubbles in pangenome graph and report super bubbles with ends present in all datasets? Is your method faster than bubble search, super bubble search?
We haven't tested that yet, but there was an idea also, not from me personally, but there was a colleague of mine who actually tried something in that direction to resolve bubbles in the graph, and by this somehow try to detect the core. And that approach, well, it became quite complicated very fast and was not that successful so far, and this is why we went for this approach. Yeah, but in the end, I cannot tell you yet whether this might not be a faster solution.
Okay. Are there any more questions? Please write them. I see something in the chat. Okay. Okay, no, I think there are no more questions. So thank you.